Okay, today we're going to be talking about the process of going from DNA into proteins, which is called transcription and translation. So first, we start off with DNA in the nucleus right here. And DNA in the nucleus is translated or transcribed from DNA into RNA. And they're both made up of nucleotides, so they're made up of the same monomer. And so after it is transcribed, an mRNA is the messenger RNA. So that's the thing that takes a message from DNA into protein. And so that's how the cell is able to know what proteins it needs to make. It's based on the genes or the sequences of nucleotides in the DNA. That allows the cell to know what protein to make. So then after it's transcribed, it comes out the nuclear pores like this. And so these are nuclear pores, these different dots in this picture. And so the mRNA comes out, and then we have a ribosome made up of rRNA, which is this thing right here. And that allows tRNA molecules, these things right here, to come in and bring the amino acids that are needed for the protein. So we have on the RNA different sequences of nucleotides, and each set of 3 is called a codon. So here's a codon right here, here's another codon, and it goes on down the line like this in sets of 3. Your start codon is always going to be AUG. And that's what allows the ribosome to come and bind to it. It recognizes that codon, and then it comes and binds, and a respective tRNA molecule will bring in the amino acid that is needed. And so the tRNA that binds with AUG is always going to have the protein methionine. So proteins will always start with methionine. And then for the next codon, another tRNA molecule will come in. And so this one, the codon, is UUC. So the anticodon is the different nucleotide bases that are on the tRNA molecule. And those are going to be complementary to the mRNA. So each tRNA will have their own anticodon right here that binds to the mRNA, and it will also have an amino acid that it brings in to attach. So we have different amino acids on here: methionine, phenylalanine, and lysine. So the tRNA molecules will always carry that same type of amino acid to the protein to help make the protein. So this one, UUU, will combine with AAA. So this tRNA will come in into the ribosome. There's an attachment site, or the A site. So the tRNA will come in, and then it will move to the P site in the middle, around here, and that's where it will attach the amino acid to the ones that are already there. So it's called the P site, or the peptidyl site. It makes a peptide bond between the amino acids. And then lastly, it'll go to the E site over here, and once the amino acid is attached, the tRNA molecule will exit from the E site and go pick up another amino acid. It will continue to do this until the protein is made. And so that is the process of going from DNA into mRNA into protein, or it's also called transcription and translation.